In this topic, we're going to discuss microorganisms in the food industry. So, by the end of the topic, you'll understand what biotechnology is, why do we use microorganisms, and what are some examples. So, what is biotechnology? Biotechnology uses microorganisms or their products to make food. For example, bacteria is used to make yogurt, fungus is used to make mycoprotein, and yeast is used to make bread and alcohol. So, yeast can be used to make bread and alcohol. How does it do this? Well, the byproducts of respiration are important. In yeast, they are alcohol and carbon dioxide. So obviously, when yeast respires anaerobically, it produces alcohol, which we use to make wine. And then if you want to make bread, the carbon dioxide is what makes the bread rise, and the alcohol will evaporate off the bread in the oven. Now we're going to look at bacteria and yogurt production. Bacteria, such as lactobacillus, is used to make milk into yogurt. The bacteria uses sugar from milk as its energy source. This sugar is called lactose. The bacteria converts the lactose to lactic acid, and lactic acid is what tastes sour and changes the pH in the milk. It causes the proteins in the milk to clump. There are other types of bacteria used. So here you can see lactose is converted into lactic acid and energy, and that lactic acid is what changes the pH of the milk so that the milk starts to clump. So milk is first pasteurized by heating it to 70 degrees Celsius to kill the disease-causing bacteria. When the milk has cooled, bacteria are added. The optimum temperature is about 40 degrees Celsius. The bacteria respire anaerobically to produce lactic acid. This slows the pH of the milk and causes it to coagulate. The yogurt is then cooled and fruit or flavoring may be added. Now the bacteria can be killed or it can be left. If it's left, we call it live culture, and it's kept cold to inhibit growth of the bacteria. In live cultures, the bacteria can help digest food in our guts, so it is quite helpful. So there's a difference between batch culture and continuous culture. Yogurt is an example of batch culture. So when yogurt is made, it's done in batches in large tanks called fermenters. This is what we call batch culture. At the end of making one batch, the fermenter is emptied and cleaned, then used again. Now we're going to look at continuous culture. Yeast or bacteria are grown in nutrients which they convert to a protein. This process is very efficient. They can be grown in fermenters continuously. Some cells are removed regularly and more nutrients added. This is what we call continuous culture. This is used to increase protein in animal feed. So notice the difference between batch culture, where you make a batch, you clean the fermenter. In continuous culture, you are continuously using the yeast in the fermenter. Some cells are removed regularly and other nutrients added. So let's have a look at how fungus is used to make mycoprotein. There's a fungus called Fusarium, and it's efficient at converting inorganic nutrients into protein. It's grown in continuous culture in fermenters where it can be continuously removed. So you have more nutrients such as ammonia and glucose are added, and the fungus continues to grow. Ammonia is a source of nitrogen for making amino acids. So the ferment is kept at 30 degrees Celsius. Now we know that Fusarium is a fungus, and this reproduces quickly and makes a massive mycelium. The fungal hyphae of the mycelium are removed and processed, then packaged. Mycoprotein is a meat substitute. It is high in protein but low in fat. So this means that it's got a lot of protein and a lot of fiber. It also has a fibrous texture due to the thread-like hyphae. It's quite bland to taste, but can easily be flavoured. So an example would be corn, which is used in Britain. Quorn. It's a brand name. So what have we learned in this topic? We've discussed biotechnology and how we can use microorganisms in industry. So what do we use to make yogurt? We use bacteria, and that's batch culture. How do we make mycoprotein? We use something called fusarium, which is a fungus, and that's using continuous culture. And that concludes our lesson. The end.